The final passive transport mechanism we're going to talk about is facilitated diffusion. Just like diffusion and osmosis, facilitated diffusion is a passive process, so there's no extra energy needed. We are just powered by the kinetic energy of the molecules involved. So as such, we are still being transported across the membrane from a high concentration down to a low concentration. That's the net movement. But remember, the molecules can go in both directions, but we just have a tendency to go from high to low concentration. It's much more rapid than simple diffusion. So much faster than simple diffusion, and we're going to see an example of facilitated diffusion moving water, which is much quicker than moving water by osmosis. We have two types of transport mechanism by facilitated diffusion. We have protein channels, and these form a channel through the membrane that allow specific materials to pass through. We can get movement in both directions, but remember, we've got a tendency to go down the gradient. So in this example here, we will be moving the green hexagons down their concentration, where we've got lots of them, to where we've got a lower amount of them. So that's through a protein channel. The other example is the carrier proteins, which is shown here. And what carrier proteins will do, again, down the concentration gradient of whatever molecule it is, but in this case, the green ovals will bind, the carrier protein changes shape, and the green oval will cross the membrane. And again, they can go in both directions. So if we look at channel proteins first in a little bit more detail, what you've got is the protein is embedded within the phospholipid membrane, and it is water-filled. It's a water-filled pore or hole through there. Therefore, materials that are soluble, that are in the water, can easily pass through the channel protein in both directions. So we'll go through here in both directions. What we will say is that they are specific. So one type of channel will only let one type of material through. So this channel will let through the green hexagons. It won't let through the green ovals. So they are specific to what they will allow to go through. Another thing that channel proteins can be is they can be gated. This means they can be turned on and off, or opened and closed, whichever way you want to look at it. So we've got a simple example here. We've got the red circles, and we've got a concentration gradient of red circles. So we've got high amounts of red circles, low red circles. We've got the channel in pink, but the channel is closed. The red circles cannot get through. What we need to operate them is we need a chemical, which we call a ligand, to bind to a site on the channel protein. This allows the channel protein to open, and now the red circles can travel down their concentration gradient through the membrane. So they can be gated. An example of a channel protein which we will meet in future weeks are aquaporins. Aqua means water, and pouring is pour for whole. And what aquaporins are is they are a protein channel in the membrane that selectively allow water through. A lot of water, very fast, very rapid, much faster than osmosis, because instead of having to squeeze through the membrane, they can just flood through the channel. And we'll find these in the nephron when we're looking as part of the kidney in the urinary system. To the right, we have a protein model of the aquaporin. So this is embedded in the membrane, so we can draw some phospholipids around it. And what we have, if we remember back to our protein structure that we talked about, we have got alpha helices through the membrane. We've got 6 alpha helices that hold this protein in position and in the correct format to allow vast amounts of water through. What carrier proteins do is a little bit different. So again, they're still passive, they're still going down their concentration gradient, but what happens is the substance joins to the protein, the protein changes shape and physically changes shape and carries the substance across the membrane. And again, they are specific. They will only allow one type of substance to cross, but they can still go in both directions, but the overall tendency is to go from high to low concentration. An example of a carrier protein involved in facilitated diffusion is GLUT1. GLUT1 is involved in glucose uptake in our cells. So what we have here is, this is outside our cells, this is inside our cells. So we've got a high concentration of glucose outside. And the reason we've got a high concentration of glucose outside is because our digestive system has taken the food we consume, broken it down into monosaccharides like glucose, absorbed it into our blood, and our blood has delivered it to all the cells in the body. And we're constantly bringing more blood with high quantities of glucose compared to the inside the cell. So the glucose is going to go down its gradient from from high to low. Remember, glucose is too big to just squeeze through the membrane. So instead, what it does is it binds to this GLUT1 transporter, shown in red. The GLUT1 changes shape, and that means that the glucose is then brought into the cell. Now we maintain this glucose gradient, we maintain the glucose as being low inside because this glucose immediately enters into aerobic respiration. So it immediately goes into glycolysis and is changed from glucose to glucose 6-phosphate. And we'll talk about that when we talk about aerobic respiration. One last thing to talk about, just to mention when we're talking about carrier proteins across the membrane, is you get different types of carrier proteins and pumps when we talk about them. GLUT1 is an example of a uniporter. Uni means one. So all it means is it just lets one material pass through the membrane. So in this case, glucose will pass down its concentration gradient. When we look at active transport in our next session, we will be seeing examples of symporters and antiporters. These are co-transporters. What this means is they carry two materials at once, either in the same direction as a symporter or in opposite directions as an antiporter. But that's something to look forward to next time. Thank you.